Good morning, students. How are you all, children? I hope all are fine. The last class I gave that one homework portion. Am I correct? Have you completed that work? Say yes or no, children? Yes. Those who didn't complete it soon. Okay? Yes. Then let us one review the previous class portion. The last class we have studied bacteria, size and shape of bacteria, ultrastructure of bacteria, and then finally learned that it's a gram staining procedure. What are the steps for gram staining procedure? Am I correct? So this much of the contents only studied in the previous class. So in this class we are going to learn the next one is what is the difference between gram positive and gram negative bacteria? Gram positive and gram negative bacteria. Okay, gram negative. What is the differences between gram positive and gram negative bacteria? First of all, so here the characteristic features. If you want to learn that's a gram positive and gram negative type of bacteria, learn that's the characteristic features of gram positive and gram negative type of bacterial cell. First one is the cell wall. So the gram positive type of the cell wall is very thick layer. Okay, the measurements also they are mentioning here. The next one, gram negative bacteria, this is very thin layer. Clear? Then rigidity of the cell wall. The second characteristic features the rigidity of the cell wall. So the gram positive type of this bacterial cell, that's a rigid. So their presence in that is the peptidoglycan molecules. Okay, why that the cells get a rigid, children? The presence of peptidoglycan molecules. Clear? Then gram negative, that is the elastic, due to presence of their lipopolysaccharides. Okay, the lipopolysaccharides and lipoproteins. So these are the two items, these are the two components is present there. So that's why the cell wall is elastic in nature. Clear? Yes. Next one is the chemical composition. What are the chemical substances present there? Means the gram positive type, the peptidoglycan is they present in a 80%. The remaining, so their presence polysaccharide is the 20%. Okay. So then also their presence in some of this techoic acid, and then what about this techochronic acid. So these are the items or their presence in the cell wall chemical substances in gram positive type. Clear? Then gram negative, so their presence in a peptidoglycan 3 to 12% only. Okay, then remaining, so their presence in the polysaccharides and lipoprotein molecules. Okay, yes. So here the techoic acid is absent. The gram negative type, the techoic acid is absent. The next one is the outer membrane. So this is the three layers they representing in that is the gram negative type. So there one layer is the presence outer membrane. The outer membrane is the gram positive type is absent. Gram negatives that is presence. Clear? The next one is periplasmic space. The periplasmic space also gram positive type that is absent, then gram negative type so there is presence. Understood, children? Yes. Next one, the nutritional requirements. The compared to that is a nutritional requirement relatively complex in gram positive type of bacteria. And comparatively, that's a relatively very simple type is they require for this nutrition is gram negative type of bacterial cell. Understood, children? Yes. The next one is flagella. Flagella gram positive type, so their presence in a 2 basal body rings. Clear? The 4 basal body ring is present in a gram negative type of bacterial cell. Understood, ma'am? Yes. Next one, lipid and lipoprotein. The lipid and lipoprotein molecule is very low amount only the presence in a gram positive type. So that's a higher amount is presence in the lipoprotein and that's a lipid content is presence in a gram negative type. The last one is the lipopolysaccharide is totally absent in gram-positive, the gram-negative that is present in this item. Understood, children? So this is one of the tabular column. What is the difference between? So that's the difference between gram-positive and gram-negative type of bacterial cell. Okay, this is very important, one of the 5-mark question, children. So all should cover this question. Clear? The next one, we are moving, that's today's class portion. Life processes in bacteria. Life processes in bacteria. Life processes in bacteria. So that is comes under in that 3 types mentioned, children. The life processes means that's a respiration. The life processes in bacteria, they are given, that's the 3 topics. The first one is respiration and nutrition. The last one is reproduction. Clear, children? Life processes in bacteria. So that is, they are given, that's the topic 3. What are the 3 topics, children? Respiration, nutrition and reproduction. Clear? First one is, we are going to see, this is the respiration. The respiration in bacteria, so here they are classified into 2 type of respiration, children. One is aerobic respiration and another one is anaerobic respiration. And another one is the anaerobic respiration. So this respiration, anaerobic means already you know that is the concept. So that for that growth and metabolic activities, the bacterial cell, the presence of this oxygen means, so that's the growth and metabolic activities are smoothly going. So the presence of oxygen. For that growth and metabolic activities, they require that is oxygen. Understood, children? So that is known as aerobic type of bacteria. Clear? This aerobic respiration. Then aerobic respiration, so that's one of the example of the species is Streptococcus. The example of the species, examples of the species, Streptococcus bacteria. Clear? This aerobic type of bacterium, it comes under in another one type is obligate aerobes. This comes under in obligate aerobes. Okay, this obligate aerobes means mostly, so this is comes under in a micrococcus species. They require for that's the growth and metabolic activity, they require that's the oxygen. Understood, children? So that is obligate aerobes. Clear, students? Say yes or no, ma? Yes. Next one is anaerobic bacterium. Anaerobic bacterium means, so in this type of this bacterium, there are absence of oxygen, but this one is that growth and metabolic activities, they need that energy. Am I correct? So that energy is, they get it from, that is a certain fermentation reaction. Clear, children? So get their energy from fermentation reaction. Understood? Yes. So this anaerobic respiration, so that's one of the best example of the species is Clostridium. That's the example of the bacterial species is the Clostridium. Clear, children? Yes. This anaerobic bacteria also they comes under in that's one of the type is the facultative anaerobes. Clear? What is meant by facultative anaerobes means, in this type, so this can grow, that's take it in a 2 types. I mean, first one, aerobic situation also they can grow in sometimes, so that one is they takes place in the anaerobic mode. Clear? The first one, aerobic means presence of the oxygen also they grow in certain condition. So they takes place in that is the energy takes from that, get it from that is the fermentation reaction. Understood? So this is the facultative anaerobes. This is the good examples of the 2 species. The example of these 2 species. First one is Escherichia coli, E. coli bacteria, and another one is Salmonella typhi. Okay, so Salmonella typhi. So these are the two examples of facultative anaerobes. What happened in this E. coli bacteria? When this facultative anaerobes of this E. coli bacterium, first one is the infection site is the abdominal abscessus region. Clear? So in that region, this bacteria first they rapidly consume all the oxygen in that region. Then this was changed into anaerobic metabolic activities. After that they produce in an anaerobic environment. Understood? Then after this step, this is the type of this bacteria, the E. coli bacteria. They allow to growing in that anaerobic type of this bacteria. What happening after this means, this anaerobic bacteria, so that on us, they cause certain diseases in human beings. Understood, children? So this is the one of this facultative anaerobic bacteria. What is happening in that abdominal abscessus region? Understood, children? What is the examples of facultative anaerobes, children? Very good. Escherichia coli, and second one is the Salmonella typhi. Understood? Good. Very good, children. The next one is, what do you mean by capnophilic bacteria? Capnophilic bacteria. So in this type of bacterial cells, for their growth and metabolic activities, they require that carbon dioxide. Okay? This type of the bacteria, for their growth and metabolic activities, they require for this carbon dioxide. This is called the capnophilic bacteria. Understood, children? This is one of the good example of this species is that Campylobacter. Clear, children? So these are the first life processes in bacteria, respiration. Respiration comes under in two types, one is aerobic and anaerobic. Aerobic also comes under in the type of obligate aerobic, and anaerobic, so that is a facultative anaerobes. What is the example of facultative anaerobes? E. coli and Salmonella typhi. And the next one is one of the very important 2-mark questions, children. What is capnophilic bacteria? They require that growth and metabolic activity, they require for carbon dioxide. Understood? That's the example of the species is a Campylobacter. Clear, children? Say yes or no, ma? Say yes or no? Yes. Very good. The next one is the life processes in bacteria, nutrition. The second one is life processes in bacteria, nutrition. Second one is nutrition. Based on the mode of nutrition. So all of you listen carefully. Based on the mode of nutrition, they are classified into mainly 2 types. Okay? Based on the classification, the mode of nutrition, that based the nutrition is in bacteria, they are classified into 2 types. One is first one is autotrophic bacteria and heterotrophic bacteria. Autotrophs and heterotrophs. What does mean by autotrophs? Prepare that. Very good. Own food. So, you are preparing that food only. Then heterotrophic means depend on another organisms. Okay, clear. So what is that major 2 types of nutrition in bacteria? Autotrophic bacteria and heterotrophic bacteria. Clear? Then this autotrophic bacteria is also they divided into 2. One is photoautotrophic bacteria and second one is hemoautotrophic bacteria. First one is photoautotrophic bacteria and hemoautotrophic bacteria. This autotrophic bacteria means they can synthesize their own food. They use that as sunlight. Clear? That is photoautotrophic bacteria. Understood, students? Hemoautotrophic bacteria means, so this cannot, that's having the chlorophyll pigments. Clear? But in this time, they obtaining that energy, they get it that energy from inorganic and organic substances. Clear? So here they presence in a chlorophyll pigments. I mean, so here there is a use of this sunlight energy, they preparing the food. So here some of the chemical substances. I mean that's inorganic, and organic molecules they used to get that, they get it that energy, they are preparing the food. Understood children? So this is a autotrophic bacterium and hemoautotrophic bacterium. Understood ma'am? Yes. Next one, the photoautotrophic bacteria is also they comes under in a two example or two types, that's a photolithotrophs and photoorganotrophs. What are the two types of photoautotrophic bacteria? Photolithotrophs and photoorganotrophs. Photolithotrophs means, all of you listen children, here the hydrogen donor molecule is the hydrogen donor molecule is the inorganic substances. Okay, the hydrogen, the electron acceptor hydrogen donor molecule is that's which one? Inorganic substances. Photoorganotrophs means, so this hydrogen donor molecule is the organic substances, organic acid or alcohols, so that one only can be utilized this one, photoorganotrophs. Clear? So this photolithotrophs becomes under two types children, one is green sulfur bacteria and another one is purple sulfur bacteria. Green sulfur bacteria means hydrogen donor is the hydrogen sulfide, H2S, okay. Then their presence in that is the chlorophyll pigment is the bacterioviridae, okay. Then what is the example of this green sulfur type of bacteria? That's a chlorobium species, chlorobium, one of the bacterial species, chlorobium, clear? Then purple sulfur bacteria, here that's a hydrogen donor molecule is the thiosulfate, that is belongs to this thiosulfate molecules. Then their presence in that is the pigments, they possess the pigment is the bacteriochlorophyll pigments. This chlorophyll they are containing in a chlorosomes, okay. This molecule is present in that is a bacteriochlorophyll. What is the example of this species is the chromatium. The chromatium is one of the example of the purple sulfur bacterium. Understood children? What is photolithotrophs? So here that's a very good. Which is molecule is getting that one? Hydrogen donor molecule is the inorganic substance. Very good. Photoorganotrophs means organic substances is hydrogen donor molecules. Understood? Yes. Next one is this photoorganotrophs, that is they comes under only one type, that's the purple non-sulfur bacterium. Okay. The purple non-sulfur bacterium, that's the best one of the example of the species is the rhodospirillum. Rhodospirillum. Clear children? Yes. Next one is the second type, hemoautotrophic bacteria. What do you mean by hemoautotrophic bacteria children? Very good. Try it. There is absence of chlorophyll pigments. Very good. Then cannot use that sunlight energy. In this time, instead of this energy, they can get it that energy from inorganic and organic compounds. Very good children. Okay, good. So the next one is, what is hemolithotrophs? Hemolithotrophs means in this type of bacteria, oxidizing inorganic substance to releasing energy. Okay, they oxidize inorganic substances, they releasing energy. For inorganic substances to releasing energy, this is the type as hemolithotrophs. Hemoorganotrophs means here the oxidizes organic substances. Here organic substances oxidized to releasing energy. Clear children? This is the two terms of this one, hemolithotrophs and hemoorganotrophs. Understood ma'am? Say yes or no? Yes. What are the examples of they comes under in a hemolithotrophs means? Can you able to say some of this inorganic compounds? Inorganic substances, very good. Inorganic compounds, very good. Sulfur, iron, hydrogen, nitrogen. So these are the examples of inorganic substance. Can you able to say that some of this organic substance, organic compounds? Very good. Methane, acetic acid, lactic acid. So these are the molecules. Clear? Yes, good. So the next one is, let us see that hemolithotrophs, what are the examples or comes under means? Sulfur bacteria, that's a bacterial species name as Diobacillus diooxidans. Okay, Diobacillus diooxidans. Iron bacteria, ferrobacillus ferrooxidans. Ferrobacillus ferrooxidans. Hydrogen bacteria, hydrogenomonas. Okay, hydrogenomonas. That is the example of the bacterial species. Nitrifying bacteria, nitrosomonas and nitrobacter. Okay, nitrosomonas and nitrobacter. So these are the examples, or they comes under hemolithotrophs. The next one is hemoorganotrophs. Already I said this one. So which substances released the energy? Very good, organic substances. Okay, very good. The next one, the example is methane bacteria. This is the methanococcus bacteria. Acetic acid bacteria, that's a species, example species is Acetobacter. Lactic acid bacteria, that's a species name is Lactobacillus. Clear, children? This is one of the very important 5-mark question. Okay, this is one of the very important 5-mark question. What is the write an account of nutrition in bacteria? Suppose they will ask this 5-mark question means, so write it. Okay. Once again, I will repeat this one. Nutrition in bacteria, mainly they are divided into 2 types. One is autotrophs and another one is heterotrophs. Clear? But then first one is the autotrophic bacteria. They are divided into 2 types. One is photoautotrophic bacteria and another one is hemoautotrophic bacteria. Clear? The next one is photoautotrophic bacterium, also they divided into 2. One is photolithotrophs and photoorganotrophs. Similarly, hemoautotrophic bacteria also divided into 2. That is hemolithotrophs and hemoorganotrophs. Clear? Then what are the types? This comes under photolithotrophs, green sulfur bacterium and purple sulfur bacterium. Photoorganotrophs, so there comes under a purple non-sulfur bacterium. Understood, children? Yes. Next one, hemolithotrophs. What is hemolithotrophs? What is hemoorganotrophs? Write it and then examples also mention it. Clear, children? Say yes or no? Yes. Next one, heterotrophic bacteria. So they depend on another organism. So they may be comes under in a parasitic mode or saprophytic, or that is the symbiotic mode of nutrition. So these are the types comes under in a heterotrophic mode of nutrition. Clear, children? Yes. Parasitic means, so there comes under in that mycobacterium. That the example of the species is the mycobacterium. Saprophytic means, so there comes under in a Bacillus mycoides. Bacillus mycoides. Okay. Another one more type also. That's a symbiotic mode. That's a rhizobium. One of this bacteria that is present in that is the leguminous crops plants. Okay. They are the root nodules. They present this item. Clear, children? So these are the heterotrophic bacteria. Sometimes they will ask that 2-mark question. Write note on heterotrophic bacteria means, so you have to be right. This is what is the types is mentioning in this heterotrophic mode of nutrition or heterotrophic mode of heterotrophic bacteria, as well as you write about that species name, bacterial species name. Clear, children? Say yes or no? Yes. Next one, the life processes in bacteria. That's the third one is the reproduction. The third one is the reproduction. The third one, reproduction. Normally, the reproduction in bacteria, that is 2 types. One is asexual reproduction and another one is sexual reproduction. Clear? Reproduction is they comes under in 2 types. One is asexual reproduction and another one is sexual reproduction. Here, the asexual reproduction in bacterial cell, so that is they comes under in 2 types. One is binary fission and another one is endospore formation. Clear? Then sexual reproduction also they comes under in 3 types. One is conjugation. Very good. Then second one is transformation and transduction. Clear, children? So the reproduction. Next, life processes in bacteria, reproduction. The reproduction is takes place in this bacterial cell. 2 methods. One is asexual and another one is sexual methods. Okay. Asexual means, so that is comes under in 2 types. One is binary fission and second one is endospore formation. Clear? Sexual reproduction also they comes under in a 3 types. What are the 3 types? Conjugation, transformation and transduction. Understood now? Yes. So first of all, we are going to learn this the asexual method of reproduction in bacterial cell. Binary fission. So here the binary fission means during that's a favorable condition, clear? This bacterial cells are they separated into 2 daughter cells or they produces into 2 daughter cells, clear? So how will you the binary fission as takes place means just imagine this is the one of this bacterial cell means this is the cell wall area, children. This is a cell wall area. So here they presence in the nucleoid. Am I correct? So this is the cell wall region. So this is the nucleoid. What will happen in this first step means first steps. So this is the nucleoid is divided into 2. So the next stage what will happen? So this is the cell wall region. So here this is a small one of the symbol meaning construction is forming in this area. So this nucleoid is divided into 2. Okay. So the nucleoid is divided into 2. The nucleoid is divided into 2. Understood, children? After what will happen? So the first steps, the nuclear material is divided into 2. Finally, we will get that 2 separate daughter bacterial cells. Okay, the 2 separate daughter bacterial cells or get it. So this is the binary fission method of asexual reproduction. Understood now? The first one, just imagine one of the bacterial cell, the nuclear material is there. First they dividing the nuclear material. After that symbol, the construction is made it in this area. Then finally we will get that's the 2 daughter cells. Okay, 2 daughter cells or get it. 2 daughter cells or get it. Understood, children? This is the binary fission of asexual method reproduction in bacteria. Next one is endospore formation. The next one of this asexual method of reproduction in bacteria, endospore formation. The endospore formation, first of all, the unfavorable conditions only. The endospore formation, the unfavorable conditions only, the bacterial cell, they produce that endospores. Clear? The unfavorable condition they produce in that is endospore. The favorable condition they can germinate and they forms that full bacteria. So what does mean by endospores? The endospores means, so this is a thick-walled resting spores are called endospores. Clear, children? The thick-walled resting spores are called endospores. Understood? This thick-walled resting spores are called endospores. So, endospores means first one, that's the unfavorable conditions only. They can produce the bacterial cells or they produce us in an endospore. What is endospore? The thick-walled resting spores. The thick-walled resting spores are called, what is that? What is that, children? Endospores. Clear? Understood now? Then favorable condition only this endospores can germinate and they forms that full form of the bacterial cell. Understood? Say yes or no? Yes. Next one is, we are moving this the second type of reproduction. This is the major type of the reproduction is the sexual type of reproduction. Sexual type of reproduction. Clear? The sexual type of reproduction is comes under 3 methods. What are the 3 methods? Very good. First one is conjugation. Then second one is transformation. And third one is transduction. Okay. The third one is transduction. Understood, children? Yes. The first one, the sexual method of reproduction in bacterial cell. Normally, the sexual reproduction means the production of and fusion of gametes, they form the eggs. Am I correct? But in this condition, the bacterial cell, the formation and the production of this spores and the formation and diffusion of this item, That's absent totally. So here only there's a gene recombination only takes place. Okay, the gene recombination. So that way only the sexual reproduction takes place in bacterial cell. Understood, children? The first method, conjugation. Conjugation. So here let's propose this conjugation that demonstrated that person, the conjugation. That person name is known as Liederbach and Edward J. Tatum. Clear? There's a two person, they demonstrated that the conjugation in reproduction, conjugation type of the asexual reproduction in bacteria, Liederbach and Edward T. Tatum. Clear? Then which bacterial cell they have choose to demonstrate this type of bacterial cell? So that's a bacterial, that's an example of the species name is known as E. coli. Okay, in the year of 1946. In the year of 1946. Clear, children? So these are the three one-worders there. The conjugation that's demonstrated by the two person, Liederbach, and another one is Edward T. Tatum. So that persons, they are demonstrated using the bacterial cell is the Escherichia coli, E. coli, in the year of 1946. Clear, children? Then, how will it take place this conjugation, reproduction, the sexual type of reproduction? How will you that's transferring the genes from one cell to another cells means? First one, this conjugation type of this sexual reproduction. So here they representing in the two bacterial cells. Okay. So first one, so this is the donor cell. This is the donor cell. This is the recipient cell. Okay. So this is the recipient cell. But donor cell, they has fertility factor. They has fertility factor. So first one, so here they present in a bacterial chromosome. Then next one is they present in a F plus factor. So that means by, so this is the item we are called that's plasmid. Okay, F plus factor. So here this bacterial cell, so only they presence in a bacterial chromosomes, but not have that plasmids. Okay, so this is the F minus cell. So that's the fertility factor is absent in this. How the gene is transferring from here to here? This is the one cell to the next bacterial, another one bacterial cell means. First one, this donor cell, they get attached to the recipient cell through that's a pili. The last class we have studied what is pili. So that's again additionally that's one type of the pili is present. That pili name is known as sex pili. Am I correct? But during this time, this conjugation time, with the help of pili only, they connect these two cells, donor cell and recipient cells. Understood, children? After that's the growth of this pili, they forms that conjugation tube. Okay, so the next step, so they forms that conjugation tubes. So of this two bacterial cells. Clear, children? So this is the two bacterial cells. They connecting in the growth of this pili. Finally, they forms that conjugation tube. Conjugation tube. Clear, children? Yes. Next one. So how will you? This is the DNA strand is they transferring from one cell to the next cell means. Here, this plasmid is double-stranded DNA structure. Clear? The first one, this double-stranded DNA molecule, this donor cell, they give that one strand of the DNA molecule is, they transfer through this conjugation tube. The recipient cell, they get it in that is the one strand of the DNA molecule is fixed. Clear? How will you? This is the double-stranded plasmid molecule. This is the double-stranded molecule. How will you transferring the genes from one bacterial cells to the recipient cell of the bacterial cell? So here, that's a one strand of this molecule is transferred through. This is the recipient cell. The recipient cell is they get it in a one strand. Understood? Then, so finally, here also one strand is there, there also one strand is there. Then the second strand, this is a double strand of DNA, no? So that's why this the second strand is they synthesized only. Understood, children? The second only, it's the recipient cell, they make it in that another one strands. Understood, children? Say yes or no? Now, what will happen in the final stage means, what will happen in the bacterial cells of this final stage means, these two bacteriums, they have plasmids. Okay? These two bacteria, they have that plasmids. Okay, children? So, this also, they have that F plus factor. This also, they have that F plus factor. So, how will you transferring that is the F fertility factor into that another one of this bacterial cell means this is the way only they transferring the genes through this method. Understood, children? Say yes or no? The first one, all of you listen now. The sexual reproduction in bacteria. The first method is the conjugation method. The conjugation, first we are selecting in the two same bacterial species. First one is donor cell and another one is the recipient cell. Donor cell, they have fertility factor, F plasmid is there. Recipient cell, there is a, without there is a fertility factor. Then the second step, so here there is a plasmid, there is a ability they have in that two stranded DNA molecule. The one strand is transferred to the recipient cell through that's a conjugation too. Understood? Then recipient cell also they get it that one strand is from there. Then after that, so this recipient cell they make it in another one strands also. Clear, children? This is the way last finally they get it in the two bacterial cells or they has that fertility factor, F plus factor. Understood, ma? Say yes or no? Yes, good, very good. So the remaining two types of the sexual reproduction we are going to see in the next class. Correct? Yes. Before winding it's my section, I will give some homework question answers, children. So all should try to learn, children. If you are learning, this is the time means you can able to easily write your public examination. Clear? Yes, very good, children. Note it. So first one is, what are the types of respiration? Aerobic and anaerobic. There is one of the important question. Write note on facultative anaerobes. The next one is one of the 2 mark question. What are capnophilic bacteria? Clear? The next one is nutrition in bacterium. They prepare one of this 5 mark question. Very, very important one. Clear? Many one words are there. They clearly you have to be go through it. The next one is very good. Reproduction. Write note on binary fission and write note on endospore. Sometimes they will ask that 2 mark question, diagram part, draw and label the parts of endospore. Clear? Then next one of the 5 mark question, very good, explain the conjugation sexual method of bacterial cell. Clear, children? Say yes or no? Thank you, students.